I have the pleasure of introducing our next speaker, Dr. Yang Hu. Dr. Yang Hu is a clinician scientist in the ophthalmology department here at Stanford. His lab focuses on the molecular mechanisms responsible for axon injury-induced neuronal degeneration. The title of his talk is Single Transcriptome Analysis of Regenerating RGCs Reveals Potent Glaucoma Neural Repair Genes.
So it's really a great pleasure to be here. ODD meeting is one of my favorite meetings. I always would like to talk something most recent funding in my life. And first of all, I'm not a clinician scientist anymore. I don't do clinical work anymore. I only focus on lab research. And this is my disclosure, which has nothing to do with the talk I'm talking about, and this is the objective. So we are firm believer of in vivo study. So we are only interested in retinal ganglion cell and optic nerve neuropathies, but we do everything in mouse eyes. So as you know, the RGC send out their axon to form the optic nerve, and anything injured of the optic nerve can induce retrograde RGC cell death. To mimic that, one of the major diseases of optic neuropathy is glaucoma. So to study glaucoma, we need a good model. To study any disease, you need to have a good model that is relevant to clinical patients. So that's why we developed a model that by mimicking human patients with the silicon oil blocking the pupil to cause the pupillary blocking, which is induce ocular hypertension to cause glaucomatous neurodegeneration. And also this model that can allow us to dilate the pupil to push away of the silicon oil bubble to allow we have a way in vivo to modulate the intraocular pressure and to generate different form of glaucoma. And of course, we also have some normal tension glaucoma, which is basically genetic modulation of the caused neurodegeneration in optic nerve and RGCs. And for ODD, that once we have a good model, so anything that we develop through other study in the mouse eyes in vivo will be applicable right away. So retinal ganglion cell and optic nerve is relatively a simple system in thin eyes. So the readout is very clear cut. The RGC cell death we can readily detectable, and the axon degeneration that can be detected clearly. And also that if we can induce the intrinsic regrowth capability of the retinal ganglion cell, we can induce axon regeneration, which can be treating through in vivo to look at that. And we can genetically modulate the RGCs using AAV-mediated method, which is, by the way, is also a gene therapy vehicle that allow us to translate what we found in the lab into clinical rather quickly. And in addition to the histology study, we need to have an in vivo readout of the RGC degeneration or survival function. So that's why we developed using the SIO to look at the morphology of RGC, and also after light stimulation to look at the response of RGC in mouse eyes in vivo. And using this way, we can really look at the individual RGCs. They're responsive to the light stimulation under different disease conditions and follow the same RGC at different time points to really study the progressive change of RGC function and in response to disease or treatment. So that's what we try to accomplish using this in vivo study, and to study these optic neuropathies that which I have elaborated that we need a good clinical relevant animal model, and we need to have a way in vivo to detect their morphology and activity. That's what I talked about before in the previous two ODD meetings. But today, I want to talk about something recently. We have an effort to look at using single cell analysis of regenerating RGC to identify the genes, the target that can be potentially as a modulator for the optic neuropathy degeneration. So as we know, optic nerve belongs to CNS. CNS axons don't regenerate in mammals. That's also true for optic nerve. If we crush the optic nerve, if we treating the axons, that we cannot detect any regenerating axons in the naive wild-type animals. However, about 14 years ago, that I involved in the study of P10 deletion can cause promote significant axon regeneration in optic nerve. So this is very exciting, and that's really open a field to study CNS axon regeneration. However, the question is why P10 knockout can cause regeneration, right? So to answer that, I guess every biologist student can come up with an idea. So since P10 knockout causes this regeneration, so we just look at the knockout RGC compared to without wild type RGC to look at the difference. Then we have a clue how that induce regeneration. So that's exactly what we thought. But before that, we know that the P10 is involved in the PI3K mTOR pathway, which is a very critical pathway in any cells that control the cell proliferation, survival. And all this question mark is that when I establish my lab, I try to answer all these questions, all these question marks by experimentally to determine their function in optic nerve regeneration. So through all these publications, we actually have a better idea in terms of these pathways that downstream of PI3K pathway, there are mTOR complex 1, mTOR complex 2 pathways. We know they have updated effect on optic nerve regeneration, and they through control of different phosphorylation site of AKT and AKT phosphorylase 473 that activate GS beta, which is inhibitory of optic nerve regeneration. But the other mTOR complex 1 pathway is really promoting regeneration. However, although we answer all these questions, we publish all this paper, but we still don't know exactly how that, what really, because P10 is a tumor suppressor gene. It activates the in general proliferation. However, how can we have a way to differentiate to only allow the regeneration happening, but don't allow activate the tumor formation pathways? So that's the question. As I elaborated before, we want to look at the difference between P10 knockout neuron and the wild type neuron. We thought we are a little bit more smarter, so we not only look at P10 knockout, because we know that there's a different capability of regeneration by gene modulation. For example, SOX3 has regeneration, but AKT3 is a little bit better than that. P10 knockout is the best single gene knockout that can induce regeneration. And then P10 SOX3 double knockout together with some other stimulator that can even generate much more strong potent regeneration. We thought if we have a way to look at all this genetic modulation, hopefully we can pinpoint the genes that cause the real gene that cause axon regeneration. We also using the ribotype mice, which is that we can purify from retinal ganglion cells specifically of the ribosome, and then purify the mRNA from ribosome to look at the gene expression profile. So we spent 3 years to cross the animals and to generate this material for sequencing. And then from different group, we really saw a bunch of genes, a group of genes that has the follow the way of regeneration capability to induce the regeneration. So we thought, okay, so that is really exciting. We have about 30, 40 genes that allow us to work on that. And then 3 postdocs spent 2 years to work on those genes and to look at their function and regeneration. Unfortunately, probably only 1 gene has very minimal regeneration phenotype, but another gene we don't see the phenotype, and although some other group published that maybe can cause some regeneration, although in our standard that's not really significant. So that's a very painful failure. So, but even more painful is because the whole idea is my idea, and there's nobody else I can complain with. And the postdoc has spent tremendous effort and on this project. So essentially, we work on this for 5 years. Just right before I moved to Stanford, recruited by Jack, and so what can we do about that? And by the way, we'll also spend at least half of our one on this. So everything is waste time and people's life. And so are we going to just stop here or not? Right? We still didn't answer the question. So the only thing good I can talk about myself is stupid, stubborn. So I still want to work on that. So what's the reason of the failure? So we have to really be honest to ourselves. Although, let's say, Peter knockout, we can have significant regeneration, but only 5% of the cell, 5% of the Peter knockout neuron can regenerate their axon. Majority, more than 90%, still not. If you look at the transcriptome from all this population mixed together, you want to find the best 5% difference from all this study. What's the chance? Of course, statistics can always give you something, right? But if from the beginning the experimental design is a failure, then that's determined to be a failure. So then we realized the only way to do that is to purify the 5% regeneration RGC, compare with in the same animal, same retina, same Peter knockout. Only difference is the regeneration capability. That's the only way to do that. That's the only way potentially be able to do that before you really you done that. So I have a hard time to convince people in my lab to do that because of this significant failure. However, there's always somebody is brave enough. This turned out is Fang Fang, which is a visiting scholar in my lab. She's a seasoned retinal surgeon. I told before her there are 2 retinal surgeons in my lab that has tried to retrograde tracing from the optic nerve to label the regenerating RTCs, but all of them failed. Fang Fang is willing to take this risk to try that, and she showed me that she is able to expose the optic nerve after optic nerve crash injury, 2 weeks after the crash injury, which is, you know, there's a lot of tissue proliferation in the blood, vessels growth there, but she can expose it beautifully. But I still don't believe she can do the retrograde tracing. And I don't know what happened. That's how difficult this project, you know, even make. That's not, yeah. Not this one. No. Okay, we have to go. So she tried different labeling dyes of proteins to be able to label the more efficiently of the regenerating RGCs. And I still don't believe her. And then until she finally showed me this figure, that in the left side is the wild type mouse eyes that after this tracing, there's nothing you can detect in the retina, which means because there's no regeneration. And in the Peter knockout mice, that there's a significant regeneration, so she can label exactly those cells by this retrograde tracing. And then two Thailand postdocs in my lab, like Liang Li, who is actually leading this project, and and Hao Liang, they work on this to optimize this another tracing method to label the total RGCs before the injury, and then to allow us to isolate the survived RGC, but don't regenerate, right? So from the same animals. And that's what they did, is that showing in the red label is the regenerating RGC in the same retina, and the green label is the survived but not regeneration. Okay, so that's if you look at the number, it's about less than 5% of compared to the survived one. And then the next difficult thing is that we have to purify those RGC and then to single cell sequencing them, which is also a challenge. As you know, the DAO retina to dissociate the cell, purify the RGC, allow them to survive, and then to do sequencing is very difficult. So that is overcome by Pei Zhuang, who is unfortunately left the lab after she developed this method, went to J&J. And so she can dissociate the develop tried mining method, and then she can dissociate the retina, get purify, fact sorting, purify the RGC, and then drop them into the plate-based so-called SMART-seq sequencing protocol, which is that we cannot do 10x, which is a more normal routine method for single cell sequencing. But first, we cannot get enough cells. Second, we cannot keep them survive before we're doing that. So that's the only way we can achieve this single cell sequencing using this way. And then the last hurdle is how to analyze those data, because the single cell sequencing, especially SMART-seq single cell sequencing result, is the analysis is very difficult, much more different than the routine 10x. And Xue Feng, who doesn't really have bioinformatics background, but she again brave enough to take on this, to learn from many different resources, get guidance from Le Cun from genetic department, who is experienced in this, to learn how to analyze those data. We know from this data analysis, we have five clusters of the RGCs mixed together from regenerating and survival RGCs. But we know their identity. So we know that in class 2 and class 4 is enriched in the regeneration RGC. And we can definitively determine the regeneration RGC identity, their subtype markers. Their majority is SPP1 expressed alpha RGC, and also have other subtype of RGC that can regenerate. So this has never been confirmed before. And then from this study, we compare regeneration and the survival RGCs, and we have a list of genes that can associate with the regeneration RGCs. Something like what we got before, right? But are they real or not? So that's the what we are more confident is because that a bunch of this top list in this top genes in this list that they involve in lipid, membrane morphology, cell migration, growth, so which is make a lot of sense of related with regeneration. So that's why we focus on this group of genes to test their function in vivo. But before that, we confirm they're enriched in this class 2 and class 4 RGCs, and immunostaining to confirm their expression in regenerating RGCs. So this is the result. So when the postdoc is doing this experiment, my heart is somewhere here, wait for the result, and until he showed me this, he said, okay, the 7 genes that we test, 6 of them has significant regeneration. So compared to the 1 gene from 40 genes that we identified before, right? So that's the powerful of the method. That's what the only way to be able to tell something is the way of the experimental design, which I learned it in a hard way. Hopefully you will doing much better work than me. Okay, so I don't know how to go back, but anyway, so the most striking protection is regeneration is comes from a gene named ANXA2, and another gene is MTP1. So we not because we want to develop a treatment, so we want to look at their function, not only in regeneration but in glaucoma model. So in the so glaucoma glaucoma model, we developed that 3 weeks in this model that has significant ganglion cell layer thickness, thinner in vivo, right? And then ANSA2 and MPP1 overexpression that can significantly increase of the thickness, prevent the degeneration. But the other 2 genes, PLIN2 and LGAL1, has less axon regeneration capability, so they have less significant neuroprotection in glaucoma. And we also did the visual function assay using OKR and the pattern ERG, and all consistent showing the ANSA2 and IPV1 has very striking protection effect. The final proof is to look at histology, right? So this is the result. In the control that you can see a dramatic RGC degeneration. However, in ANSA2 and MPP1 overexpression has very striking neuroprotection compared to the other 2 genes that are not that potent in regeneration. So that's further confirmed that the regeneration capability is somehow related with neuroprotection, which we have never known before. This is the first time we have this concept comes into mind. And again, the axon protection is also similar, consistent, striking as well. And this is the quantification. Okay, so if I have another 2 hours, I can tell you a little bit more about how this gene working downstreamly. But I think I already spent too much time of your time. So I just want to give you an idea. So this a powerful strategy, if you design it well, execute it well, it's really very powerful, can give you a lot of information that you can never get before. And hopefully I convinced you that the novel regeneration associated genes that we identified are potential neural repair candidates that has striking neuroprotection effect. And finally, I believe this method can be applicable in a more broad CNS neurodegeneration disease ways. All right, so I think I already highlight those people that I have to say I'm really lucky, fortunate to work with them. They are brave enough. They overcome a lot of technical difficulties, establish all this complete strategy to answer this question. And I want to, fortunately, we also have a lot of collaborators throughout the department and the university. I want to point out Dr. Stanley Qi from bioengineering and Dr. Le Cong is really helping us to analyze those data. And finally, all this funding that allow us to do this very expensive experiment, including all this failure, and including the funding from the ODD that we really appreciate. Thank you very much.
Thank you, Dr. Hu. Does anyone have any questions?
Hi, Dr. Hu. Amazing, amazing discovery and huge lesson to learn for a young investigator like me. So are you trying to develop a gene therapy using ANXA2 and MPP1, or have you tried any chemical agonist also of these genes, like whether they'll work so that you are more closer to the clinic?
Yeah, that's definitely our goal for these studies, right? ANSA2, MPP1, they are both, and some bunch of other genes I don't have time to talk about. Actually, they have a common feature is they involve in membrane. They locate close to membrane, convey the communication between extracellular matrix and the intracellular cytoskeleton. So I think by identifying the true mechanism of how they modulate this membrane structure, will give us a very good hint. But for this gene, in terms of ANSA2 itself, is already very striking protection effect, and we actually found the ligand of that ANSA2 and the downstream molecule of ANSA2 both have very significant axon regeneration and the neuroprotection effect. So we think that is very worthwhile to further develop as a treatment.
Awesome. Yeah. Thank you so much.
Yeah.
Thank you so much for your amazing talk. I have a question. Have you ever examined the function of the regenerated cells? Have you evaluated them functionally? And then how do you evaluate if they successfully make a synapse with the second order neurons?
secondary.
Yes.
Okay, yeah.
Thank you.
Yeah, that's very good question. That's definitely what we want to do. I showed at the very beginning that the calcium imaging that will allow us to study in vivo of their regenerating axons, but regenerating RGC. But why we didn't do that? Because currently the SIO we have is only one channel. We can look at the calcium signal, but we cannot label retrogradely of the regenerating RGC. We don't know which one is the regenerating one. Right. So if we, with Jeff's support, if we get two channels of SLO, we can label this regenerating RGC, then we can potentially to know exactly which cell is regenerating, then we can look at that. Another postdoc in my lab is working on the fiber photometry that look at their axon activity after light induced in the regenerating RGCs. So that's not secondary yet, but until we can have the regeneration long enough into the brain to connect. Actually, we saw some regenerating long enough into the brain, although they are not in the correct position, which we don't understand yet. But that's something we can potentially to look at.
Hi, Dr. Hu. Sorry, another question. Does this NXA2 and MPP1 knockout work for human RGCs also?
Oh, human RGC.
Yeah.
Oh, that I don't know. So unfortunately, I didn't work on human samples, but
Jeff has ACD tomato RGC cell line. Just try. It'll be really nice.
For sure. Yeah.
Yeah. Thank you so much.
I definitely need to move on to human. Now, thank you very much. Appreciate it.